Hello friends, good evening. Welcome to Sam Biology class. Today we shall discuss about gel electrophoresis. Before that, I request all of my friends, please subscribe my YouTube channel. Gel electrophoresis is a simple, rapid and sensitive analytical tool widely used for the separation of charged molecules. Under the influence of an electric field, charged particles migrate in the direction of the electrode bearing opposite charge. These particles have different electrophoretic mobility due to differences in their shape, size and net charge. Molecules having different electrophoretic mobility migrate at different rate through the gel matrix and are separated into distinct bands. In this technique, gels serve as the supporting medium. It acts as a molecular sieve. The gels provide a porous mechanical support for the movement of charged molecules, thereby stabilizing the medium. Due to the presence of the gel, the adverse effect of the convection current and the diffusion is minimized relative to the free electrophoresis. Different types of gel that are used in electrophoresis include number 1 starch, number 2 agarose, and number 3 polyacrylamide, and number 3 polyacrylamide. First one is starch gel. First one is starch gel. The use of starch as the stabilizing medium was introduced by Smiths in 1955. One of the major drawbacks of starch gel is that the pore size cannot be controlled. It is also difficult to prevent the contamination of starch gel by microorganisms. In addition to this, upon staining, the starch gel becomes opaque, thereby making direct photoelectric examination of the gel impossible. However, starch gel have a higher resolving power which can be matched only by the polyacrylamide gel. Initially, starch gel was used in electrophoresis, but these days either agarose or polyacrylamide are widely used for the separation of charged particles or molecules. Second one is agarose gel. Agarose is a linear polysaccharide with an average relative molecular mass about 12,000. It is made up of the basic repeat unit agarobiotes, which consists of alternating unit of galactose and 3,6-anhydrogalactose. 3,6-anhydrogalactose. Agarose is a component of agar, which is a mixture of polysaccharides that has been isolated from certain seaweeds. The porosity of agarose gel is controlled by the initial concentration of agarose. Large pore sizes are formed from low concentration, whereas small pore sizes are formed from high concentration of agarose. Agarose gel electrophoresis is routinely used for the qualitative as well as quantitative characterization of nucleic acids. It can also be used for the preparative purpose as the DNA band can be excised later on and can be purified for further use. Because of larger pore size, agarose gels are most suitable for the separation of nucleic acids. However, it is also useful for the study of protein where separation is size dependent, as in case of flatbed isolated focusing or immunoelectrophoresis. Third one is polyacrylamide gel. Polyacrylamide is a polymer of acrylamides. Cross-linked polyacrylamide gels are formed from the polymerization of acrylamide monomers in the presence of a smaller amount of N methylene bisacrylamide. A molecule of bisacrylamide is composed of two molecules of acrylamides joined by methylene group. It is used as a cross-linking agent. Acrylamide monomers are polymerized in a head-to-tail fashion into long chains. Occasionally, a molecule of bisacrylamide is introduced into the growing chain, thereby establishing a second site for chain extension. Polymerization in this way results in the formation of a cross-linked matrix with a fairly well understood structure. The pore size in the gel can be controlled by changing the concentration of both acrylamide and bisacrylamide. Acrylamide gels are made with a content of between 3% and 30% acrylamide. Low percentage gels have large pore size and are used in the electrophoresis of proteins where free movement of proteins by electrophoresis is required without any noticeable frictional effect. For example, in flatbed isolatory focusing or in the stacking gel of SDS polyacrylamide gel. Low percentage acrylamide gels are also used in the separation of nucleic acids. Acrylamide gels with a content between 10% and 20% acrylamides are used in SDS polyacrylamide gel electrophoresis where the smaller pore sizes introduces a molecular sieving effect that aid in the separation of proteins based on their molecular size. Based on the orientation of the gel electrophoresis is divided into two types such as horizontal gel electrophoresis and vertical gel electrophoresis. First one is horizontal gel electrophoresis that requires casting gel in a horizontal orientation and placing it in a chamber filled with a running buffer solution. In this technique, electrophoresis is performed in a continuous fashion with both electrodes and the gel cassette remain submerged within the running buffer. Two platinum electrodes placed at both ends are connected to an external power supply from a power pack that supply the stabilized direct current. In this technique, the running buffer solution also serves to cool down the gel as it heats up during electrophoresis. After placing different samples in the sample application wells, when the current is supplied, then the charged sample components migrate in a horizontal direction. Due to the nature of the horizontal setup, it is not feasible to use a discontinuous buffer system. The gel used in horizontal electrophoresis is generally made of agarose, which has larger pores and therefore widely used in the separation of nucleic acids. Acrylamide gel is not used in this system because it cannot polymerize in the presence of oxygen. Second one is vertical electrophoresis that involve casting gel in a vertical orientation. This technique employs a discontinuous buffer system having two buffer reservoirs. The cathode is placed in the upper and the anode is in the lower buffer reservoir. During electrophoresis, a small amount of buffer travels from upper to the lower buffer reservoir only through the gel. This allows for precise control of voltage gradient. This, along with small pore size in the polyacrylamide gel, makes this technique highly suitable for the separation of mixture of proteins. Several proteins of biological importance consist of more than one polypeptide chain. Such proteins are called oligomeric proteins, in which the subunits are held together by hydrogen bonds, disulfide bonds, and hydrophobic interaction. During electrophoresis, such proteins migrate as a single band. But if one has to study the individual subunits separately, then a class of compound called solubilizers, are used. Examples are urea, sodium dodecyl sulfate, sodium dodecyl sulfate, SDS, and beta mercaptoethanol and beta mercaptoethanol. Solubilizers destabilize the quaternary structure of proteins, and this results in the separation of individual subunits that migrate as distinct bands through the gel during electrophoresis. In this type of vertical electrophoresis, the gel is made in the form of a slab. So such type of technique is also called slab gel electrophoresis. These are the sample application wells which can be made by placing a cone-shaped jig into the gel before it sets or polymerizes. Because of the presence of several wells, several samples can be loaded simultaneously and can be studied under identical conditions. This is one of the major advantages of slab gel electrophoresis. The pH of the buffer solution is adjusted to 9 so that all sample components will get net negative charge. During electrophoresis, all of them will migrate to the anode. After application of the sample, when the electric current is switched on, charged particles or molecules will migrate vertically downwards at different rate based on their electrophoretic mobility, and as a result they will be separated into distinct bands. The sample is usually mixed with a tracking dye. This is necessary in order to track the progress of electrophoresis. Dye particles migrate at a faster rate than all macromolecules that are present in the sample. For example, if the tracking dye has just come out of the gel, or before that, if we will stop electrophoresis, then we can be reasonably sure that all sample components are present within the gel. After separation, next step is sample detection, recovery and estimation. If the gel is inside a column, then it is removed by forcing water from a hypodermic syringe around the wall of the column. In case of slab gel electrophoresis, the gel is removed by separating two gel plates. As in case of paper chromatography, in this technique also the sample components are usually detected in situ. Physical properties of biomolecules such as fluorescence, UV absorption and radioactivity in case the sample components are radioactive can be successfully exploited for their detection. Alternatively, the gel can be stained with a suitable reagent. Prior to staining, the gel can be treated with a fixative in order to prevent the diffusion of the result component. Upon staining, the starch gel turns opaque, thereby making it difficult for direct densitometry. This difficulty can be obviated by treating the starch gel with glycerol and acetic acid If one has to estimate the reserved components accurately, then the compounds are eluted from the gel and then estimated by spectrophotometry. Applications of gel electrophoresis. Apart from the separation of proteins and many other macromolecules, gel electrophoresis has several other applications. First one is determination of DNA sequence. Second one is northern and southern blotting. Third one is restriction mapping of DNA. Third one is restriction mapping of DNA. And fourth one is determination of subunit stoichiometry. stoichiometry. And finally, determination of molecular weight. Determination of molecular weights of different proteins. So friends, this is all about the principle, procedure and applications of gel electrophoresis. For more updates, subscribe my YouTube channel Sam Biology Class. Goodbye, good night.